Tonight's lecture is over Chapter 5, Energy. And upon the completion of this chapter, our learning objectives are to be able to describe how energy flows from the sun and through all life on Earth. We will be able to discuss how photosynthesis uses energy from sunlight to make food. We will be able to explain how cellular respiration converts food molecules into ATP. ATP is a universal energy source. And we will also be able to describe alternative pathways to energy acquisition. Fast food for your car? Yes. The idea isn't as far-fetched as it sounds. In fact, many vehicles already run on biofuels produced from plant and animal products. The production of biofuels requires only the plant or animal source, sunlight, air, water, and a relatively short amount of time. A few months or years, depending on the source. In contrast, fossil fuels such as gasoline, oil, natural gas, and coal are produced from the decayed remains of plants and animals modified over millions of years by heat, pressure, and bacteria. It turns out that biofuels, fossil fuels, and the food fuels that supply energy to most living organisms are chemically similar. Energy from the sun is the source of the energy stored in the chemical bonds between the atoms in all these fuels. They contain chains of carbon and hydrogen atoms bound together, and breaking these bonds and forming new, lower energy bonds releases large amounts of energy and water and CO2. So, are we at the point where our cars run only on biofuels? Not quite. Growing crops for biofuel reduces large amounts of fossil fuel and water. This is why the search for better fuels continues. Still, the use of biofuels has been increasing, and in some cases they have been incorporated into fossil fuels. Most gasoline sold in the United States, for example, contains some ethanol that is produced from corn. Moving on. In this chapter, we explore how plant, animal, and other living quote-unquote machines run on energy stored in chemical bonds. Nearly all life depends on capturing energy from the sun and converting it to forms that living organisms can use. This energy capture and conversion occurs in two important processes that mirror each other. One is photosynthesis. Photosynthesis is the process by which plants capture energy from the sun and store it in the chemical bonds of sugar. And secondly, there's cellular respiration. Cellular respiration is the process by which all living organisms release the energy stored in the chemical bonds of food molecules and use it to fuel their own lives. From the sun to you in just 2 steps. And that brings us to our first take-home message for this chapter. The energy from sunlight is stored in the chemical bonds of molecules. When these bonds are broken, energy is released regardless of whether the bond is in a molecule of food, of a fossil fuel, or of a biofuel such as the oil in which French fries are cooked. Moving on, batteries not included. For a child, those are pretty depressing words. We know that many toys and electronic gadgets need energy, usually in the form of batteries. Generating ringtones, lights, and movement requires energy. The same is true for humans, plants, and all other organisms. They need energy for their activities, from moving to reproducing to thinking. Energy is the capacity to do work. Work is anything that involves moving matter against an opposing force. In the study of living things, we encounter 2 types of energy, kinetic and potential. Kinetic energy is the energy of motion, for example, legs pushing bike pedals and birds flapping wings. Heat, which results from lots of molecules moving rapidly, is another form of kinetic energy. Similarly, because light comes from the movement of high-energy particles, it too is a form of kinetic energy, probably the most important form of kinetic energy on Earth. Moving on, an object does not have to be moving to have the capacity to do work. It may have potential energy. Potential energy is stored energy that results from an object's location, position, or composition. Water behind a dam, for example, has potential energy. If a hole is opened in the dam, the water can flow through and perhaps spin a water wheel or turbine. A concentration gradient, which we discussed in Chapter 4, also has potential energy. If molecules in an area of high concentration move toward an area of lower concentration, the potential energy of the gradient is converted into the kinetic energy of molecular movement, and this kinetic energy can do work. Chemical energy, the storage of energy in chemical bonds, is also a type of potential energy. Because potential energy doesn't involve movement, it is a less obvious form of energy than is kinetic energy. A strawberry has potential energy, as does any other type of food. Why? Because during cellular respiration, the chemical energy stored in the chemical bonds make up the food you eat, and it can be released, and this can enable you to run, play, work, think, etc. We explore cellular respiration, the energy-releasing breakdown of molecules, later in this chapter. But first we need to know more about the nature of energy. And that brings us to our take-home message for Section 5.2. Energy, the capacity to do work, comes in two forms: kinetic energy, which is the moving of objects, and potential energy, such as chemical energy, is stored energy that results from the location, position, or composition of an object. Moving on. Every minute of every day, even during cloudy days, the sun shines brightly, releasing tremendous amounts of energy. Organisms on Earth cannot capture all of the sun's energy that reaches the surface of the Earth. Indeed, most plants capture only a tiny fraction of the available energy. What happens to the rest? This unused energy does not simply disappear. All energy from the sun that reaches the Earth can be accounted for. Some, probably less than 1%, is actually captured by organisms and transformed into usable chemical energy through photosynthesis. The rest is reflected back into space, probably about 30%, or it is absorbed by land, the oceans, and the atmosphere, which accounts for about 70%, and mostly transformed into heat. Moving on, the same accounting can be used on a smaller scale. If you eat a bowl of rice, some proportion of the chemical energy stored in the bonds of the molecules that make up the rice grains is transformed into usable energy that can fuel your cell's activity. All the rest of the energy is transformed into heat and is ultimately lost into the atmosphere. Similarly, in automobiles, about 3/4 of the energy in gasoline is lost as heat. Another way to say this is that energy can change form but never disappear. This is an important feature of energy in the universe. In all our eating and growing, driving and sleeping, we are simply transforming energy. The study of the transformation of energy from one type to another, such as from potential to kinetic energy, is called thermodynamics. The first law of thermodynamics states that energy can never be created or destroyed. It can only change from one form to another. Moving on, the second law of thermodynamics states that no conversion of energy is perfectly efficient. Each conversion invariably includes the transformation of some energy into heat. Although heat is certainly a form of energy, it is almost completely useless to living organisms for fueling our cellular activity because it is not easily harnessed to do work. The second law of thermodynamics tells us that little by little, the amount of energy available to do work decreases. Another way of looking at the second law of thermodynamics is to recognize that the disorder of a closed system, referred to as entropy, always increases. And that brings us to our take-home message for Section 5.3. Energy is neither created nor destroyed, but can change form. Each conversion of energy is inefficient, and some of the usable energy is converted to heat, a less useful form of energy. Moving on, Much of the work that cells do requires energy, but even though light from the sun carries energy, as do molecules of sugar, fat, and protein, none of this energy can be used directly to fuel chemical reactions in cells of organisms. The energy first must be captured in the bonds of a molecule called adenosine triphosphate, or we will frequently call this molecule ATP. This is a free-floating molecule found in cells that acts as a rechargeable battery, temporarily storing energy that can then be used for cellular work in plants, animals, and all other organisms on the earth. ATP is a simple molecule with three components. At the center of the ATP molecule is a small sugar molecule, which is a ribose, attached to a molecule called adenine and a chain of three negatively charged phosphate groups, hence the tri in triphosphate. These molecules are held together with slightly unstable bonds that contain a large amount of energy. The instability of these high-energy bonds means that the three phosphate groups are like a tightly coiled spring. With the slightest push, one of the phosphate groups will pop off. In this process, a little burst of energy will be released that the cell can use. Moving on, each time a cell expends one of its ATP molecules to pay for an energetically expansive reaction, a phosphate is broken off. What is left is a molecule with two phosphates called ADP, that is adenosine diphosphate, and a separate phosphate group labeled P sub I. Because ADP has less energy stored in its bonds, the reaction is accomplished by a release of energy. An organism can use ADP, a free-floating phosphate, and an input of kinetic energy to rebuild its ATP stocks. When we discuss photosynthesis, we'll see that plants, algae, and some bacteria can use light energy directly from the sun to make ATP from ADP and free-floating phosphate groups. In addition, these photosynthetic organisms can use the energy generated from breaking down sugar and other molecules. That's the same energy source that animals use, generating ATP from the energy contained in the bonds of their food molecules. So whether it comes from the sun or from the breakdown of molecules such as sugar, the energy is used to recreate the unstable bond in the triphosphate chain of ATP. When energy is needed again, the organism can release it by breaking the bond holding the phosphate group to the rest of the molecule, which once again releases energy. And that brings us to our take-home message for section 5.4. Cells store energy in the bonds of ATP molecules. This potential energy can be converted to kinetic energy and used to fuel life-sustaining chemical reactions. In other reactions, inputs of kinetic energy are converted to the potential energy of the energy-rich but unstable bonds of ATP molecules. Moving on, watching a plant grow over the course of a few years can seem like watching a magic trick. Of course it's not, but the process is nonetheless amazing. Consider that in 5 years, a tree can increase its weight by 150 pounds, and after more than a century of growth, a mature saguaro cactus may weigh 3,000 pounds. Where does that 150 pounds of new tree come from? Our first guess might be the soil. It's easy enough to weigh the soil in a pot when at first planting a tree. 5 years later, however, if we weigh the soil again, we find that it has lost less than a pound, nowhere near enough to explain the massive increase in the amount of plant material. Perhaps the new growth comes from the water provided to the plant. Wrong again. Although the older and much larger tree holds more water in its many cells, rain and groundwater do not account for the increase in the dry weight of the plant. Most of the new material in a growing plant actually comes from an element present in an invisible gas in the air. During photosynthesis, Plants capture this element present as carbon dioxide gas in the atmosphere and use the energy from sunlight along with water and small amounts of chemicals usually found in soil to produce solid, visible, and often tasty sugars and other organic molecules. The molecules are used to make plant structures such as leaves, roots, stems, flowers, fruits, and seeds. In the process, the plants give off oxygen, a byproduct that happens to be necessary for much of the life on Earth, including all animal life. So let me give you a little perspective on this. Plants basically grow out of thin air, and the sugars that plants form are likely in part due to the exhaling that you, me, your dog, your cat, etc. may have done. Moving on, it should also be pointed out that plants are not the only organisms on the earth capable of photosynthesis. Some bacteria and many other unicellular organisms, along with kelp and other multicellular algae, are also capable of using the energy in sunlight to produce organic materials. Moving on, these are three inputs to the process of photosynthesis: light energy from the sun, carbon dioxide from the atmosphere, and water from the environment. From these three inputs, the plant produces sugar and oxygen. Photosynthesis is best understood as two separate events, a quote-unquote photo segment, during which light is captured, and a quote-unquote synthesis segment, during which sugar is built. In the photo reactions, light energy is captured and temporarily saved in energy storage molecules. During this process, water molecules split and produce oxygen. In the synthesis reactions, the energy in the energy storage molecules is used to assemble sugar molecules from carbon dioxide from the atmosphere. And that brings us to our take-home message for section 5.5. During photosynthesis, plants use water, sunlight, and carbon dioxide gas to produce sugars and other organic materials. Photosynthesizing organisms also produce oxygen, which makes all animal life possible. Moving on, if a plant part is green, then it is photosynthetic. Leaves are green because the cells near the surface are packed full of chloroplasts. Chloroplasts are light-harvesting organelles that enable the plant to use the energy from sunlight to make sugars and plant tissues. Other plant parts, such as stems, may also contain chloroplasts, in which case they too are capable of photosynthesis, but most chloroplasts are located within the cells of the leaves. Moving on, chloroplasts are sac-shaped organelles filled with a fluid called stroma. Floating in the stroma is an elaborate system of interconnected membranous structures called thylakoids, which are ordered in groups that often look like stacks of pancakes. Embedded within the thylakoid membranes are many light-capturing pigments organized into structures called photosystems. Inside the chloroplasts, there are two distinct regions, inside the stroma or inside the thylakoids. The conversion of light energy to chemical energy, the photo part of photosynthesis, occurs on the surface of and inside the thylakoids. The production of sugars, the synthesis part of photosynthesis, occurs within the stroma. And that brings us to our take-home message for section 5.6. In plants, photosynthesis occurs in chloroplasts, green organelles packed in cells near the plant's surfaces, especially in the leaves. Moving on, you can't eat sunlight. That's because sunlight is light energy rather than the chemical energy found in the bonds of food molecules. Photosynthesis is powered by light energy, a type of kinetic energy made up of little energy packets called photons, which are organized into waves. Photons can do work as they bombard surfaces such as your face, and in that case it will heat your face, or a leaf, which in that case it enables the leaf to build sugar from carbon dioxide and water. Moving on, photons have various amounts of energy, and the length of the wave in which they travel corresponds to the amount of energy carried by the photon. The shorter the wavelength, the more energy the photon carries. Within a ray of light, there are super high energy photons, those with short wavelengths, relatively low energy photons, those with longer wavelengths, and everything in between. This range, which is called the electromagnetic spectrum, extends from extremely short, high-energy gamma rays and X-rays with wavelengths as short as 1 nanometer to very long, low-energy radio waves with wavelengths as long as a mile. Just as people cannot hear some high-pitched frequencies of sound that many dogs can hear, there are some wavelengths of light that we cannot see. The light that we can see is called visible light, and it spans all the colors of the rainbow. Humans and other animals can see colors because our eyes contain light-absorbing molecules called pigments. The energy in light waves excites electrons in the pigments, stimulating nerves in our eyes, which then transmit electrical signals to our brain. We perceive different wavelengths within the visible spectrum as different colors. The pigments in the human eye absorb many different wavelengths pretty well, and that's why we can see so many different colors. Moving on, when plants use sunlight's energy to make sugar during photosynthesis, they also use the visible portion of the electromagnetic spectrum. Unlike the pigments in our eyes, however, plant pigments, the energy-capturing parts of a plant, absorb and use only a portion of the visible light wavelengths. Chlorophyll is the main pigment molecule in plants that absorbs light energy from the sun. Chlorophyll molecules are embedded in the thylakoid membranes of chloroplasts, just as light energy excites electrons in the pigments responsible for color vision in humans. Electrons in a plant's chlorophyll can become excited by certain wavelengths of light and can capture a bit of this light energy. Plants produce several different light-absorbing pigments. The primary photosynthetic pigment, called chlorophyll A, absorbs red and blue-violet wavelengths of light. Every other wavelength generally travels through or bounces off this pigment. Chlorophyll A cannot efficiently absorb green light, but instead reflects those wavelengths. We perceive the reflected light waves as green, so the pigment and the leaves contained in it appear to be green. Another pigment, called chlorophyll B, is similar in structure to chlorophyll A, but absorbs blue and red-orange wavelengths, and it reflects yellow and green wavelengths. Related pigments called carotenoids absorb blue-violet and blue-green wavelengths and reflect yellow, orange, and red wavelengths. Moving on, in the late summer, cooler temperatures cause some trees to prepare for the reduced daylight of winter by shutting down chlorophyll production, which is energetically expensive. This, in turn, reduces photosynthetic rates and causes a slowdown, much like an animal's hibernation. Gradually, the chlorophyll A and B molecules in the leaves are broken down and their chemical components are stored in the branches. As the amounts of chlorophyll A and B in the leaves decrease relative to the remaining carotenoids, the striking colors of the fall foliage are revealed. During the rest of the year, chlorophylls A and B are so abundant in leaves that green masks the color of the other pigments. After the cold winter, the increase in daylight and in temperature signal the plant to again produce chlorophyll and new leaves. And that brings us to our take-home message for section 5.7. Photosynthesis is powered by light energy, a type of kinetic energy made of energy packets called photons. Photons collide with chlorophyll and other light-absorbing molecules in chloroplasts of cells near the green surfaces of plants. These molecules capture some of the light energy from photons and harness it to build sugar from carbon dioxide and water. Moving on, An organism can use energy from the sun only if it can convert the light energy of the sun into the chemical energy of the bonds between atoms. The most important molecule in this conversion is the pigment chlorophyll. When photons of certain wavelengths hit chlorophyll, the light energy bumps an electron in the chlorophyll molecule up to a higher energy level, in other words, an excited state. Upon absorbing the photon, the electron briefly gains energy and the potential energy in the chlorophyll molecule increases. Moving on, the excited electron generally has one of two fates. First, the electron can return to its resting unexcited state, releasing energy in the process, some of which may bump electrons in a nearby molecule to a higher energy state, while the rest of the energy is dissipated as heat. Alternatively, the excited electron itself can be passed to another molecule. The passing of electrons from molecule to molecule is an important way that energy moves through cells. Many molecules carry or accept electrons during cellular activities. All that is required is that the acceptor must have a greater attraction for the electrons than does the donor molecule. A molecule that gains electrons always carries greater energy than it did before receiving the electrons. For this reason, the passing of electrons from one molecule to another can be viewed as a passing of potential energy, which is one of the first steps in photosynthesis. Because particles in the atmosphere can block light from the sun and reduce the excitation of electrons in chlorophyll molecules, photosynthesis can be hindered by light-blocking particles in the atmosphere. Any reduction in the available sunlight can have serious effects on plants and the animals that eat them. Scientists hypothesize that this is how the dinosaurs were wiped out about 60 million years ago. At that time, a large meteor hit the Earth, and around the same time, there was massive volcanic activity. Smoke, soot, and dust in the atmosphere from these events may have blocked sunlight to such an extent that many plants in the region of the impact and beyond could not conduct photosynthesis at high enough levels to survive. When plants die off, the animals and other species that rely on plants for energy die as well. Nearly all life on Earth is completely dependent on the continued excitation of electrons by sunlight. And that brings us to our take-home message for section 5.8. When chlorophyll is hit by photons, the light energy excites an electron in the chlorophyll molecule, increasing the chlorophyll's potential energy. The excited electrons can be passed to other molecules, moving to potential energy through the cell. Moving on, photosynthesis is a complex process, but our understanding can be greatly aided by remembering one phrase: follow the electrons. Where are they coming from? What are they passing through? Where are they going? What will happen to them when they get there? In the photo part of photosynthesis, sunlight hits the chloroplasts of a plant's leaves, and some of the energy in this sunlight is captured and stored in ATP and in another molecule called NADPH, which stores energy by accepting high-energy electrons. After these transformations, the captured energy is ready to be used to make sugar molecules in the synthesis part of photosynthesis. The energy capturing process occurs in two types of photosynthesis that are linked and work together. Embedded in the thylakoid membranes in the chloroplasts, these photosystems are structures composed of light-capturing pigments, including chlorophyll and proteins. As the pigments absorb photons, electrons in the pigments gain energy and become excited, and then return to their resting state, releasing energy. The released energy, but not the electrons, is transferred to neighboring pigment molecules. This process continues until the energy transferred among many pigment molecules makes its way to a chlorophyll A molecule at the center of the photosystem and excites an electron there. The chlorophyll A molecule at the center of the photosystem differs from the other pigment molecules when its electrons are boosted to an excited state. They do not return to their resting, unexcited state. Instead, a nearby molecule called the primary electron acceptor acts like an electron vacuum, grabbing the excited electrons and leaving electron vacancies. This is where the electron journey begins. Moving on, the electrons taken away from the special chlorophyll A molecule at the center of any photosystem must be replaced for photosynthesis to continue. Water supplies the replacement electrons in the first of the two linked photosystems. Molecules of water are continuously split near the special chlorophyll A molecule in this first photosystem. 4 photons of light split 2 molecules of water into 4 electrons, 4 protons, and a molecule of oxygen. A convenient and life-sustaining byproduct of the splitting of water in photosynthesis is oxygen. The oxygen released from the cell through this process is essential for much of the life on Earth, including all animal life. Moving on, once the primary electron acceptor in the first photosystem grabs a high-energy electron from chlorophyll A, it passes it to another molecule and so on. This process is called an electron transport chain, and it links the first and second photosystems. The electron transport chain is embedded within the thylakoid membrane and consists of several protein complexes that hand off electrons from one to the next. At each step in the electron transport chain sequence of handoffs, the electrons fall to a lower energy state and energy is released. These bits of energy are harnessed to power pumps in the thylakoid membrane that move protons, also referred to as hydrogen ions, from the stroma to inside of the thylakoid. Moving on, as protons are pumped into the center of the thylakoid sac, their accumulation leads to higher and higher proton concentrations in that space. Think of a pump pushing water into an elevated tank, creating a store of potential energy that can gush out of the tank with great force and kinetic energy. Similarly, the protons eventually rush out of the thylakoid sacs through the molecule ATP synthase with great force, and the force of the protons moving down their concentration gradient is harnessed to build energy-storing ATP molecules from ADP and free-floating phosphate groups. This ATP is one of 2 products of the photo portion of photosynthesis. As traveling electrons continue their journey, they reach the reaction center of the second photosystem. Like the first photosystem, the second photosystem has numerous pigments that are harnessed photons from the sun and pass the light energy to another special chlorophyll A molecule. The special chlorophyll A molecule at the center of this second photosystem has electron vacancies because, as in the first photosystem, as its electrons are boosted to an excited state, they are whisked away from the chlorophyll molecule by another primary electron acceptor. The electrons must be replaced, but rather than being supplied by the splitting of water, the electrons in the second photosystem are passed from the first photosystem. The electron acceptor at the center of the second photosystem passes the electrons to a second electron transport chain. In this second electron transport chain, as the electrons are handed off, there is no pumping of protons as in the first electron transport chain. Instead, energy is harnessed as the electrons are passed to a molecule of NADP+, creating NADPH, which is a high-energy electron carrier. NADPH is the second important product of the photo portion of photosynthesis. So to this point, we've captured light energy from the sun and converted it to the chemical energy of ATP and the high-energy electron carrier NADPH. But we haven't made food. In the next section, we will discuss how plants use energy in ATP and NADPH to produce sugar from carbon dioxide during the synthesis part of photosynthesis. And that brings us to our take-home message for this section. And that is, in photosynthesis, the photo part, light energy, is transformed into chemical energy while splitting water molecules and producing oxygen. The energy of sunlight is first captured when an electron in a chlorophyll molecule is excited. As this electron is passed from one molecule to another, energy is released at each transfer, some of which is used to build the energy storage molecules ATP and NADPH. Moving on, the synthesis part of photosynthesis takes place in a series of chemical reactions called the Calvin cycle. Just as a magician seems to make a rabbit appear from thin air, the Calvin cycle takes invisible gaseous molecules of carbon dioxide from the atmosphere and uses them to assemble molecules of sugar, sufficiently numerous that we can see them as tangible, solid, even edible material. All the Calvin cycle reactions that convert carbon dioxide into usable sugar occur in the stroma of the leaves' chloroplast outside the thylakoids. Plants carry out these reactions using the energy stored in the ATP and NADPH molecules that are built in the photo portion of photosynthesis. This dependency links the light-gathering photo reactions with the sugar-building synthesis reactions of photosynthesis. Moving on, the processes in the Calvin cycle occur in 3 steps. First, fixation. Using an enzyme called rubisco, plants pluck carbon from the air where it occurs in the form of carbon dioxide, which has 1 carbon, and attach or quote-unquote fix it to another organic molecule, which has 5 carbon atoms within the chloroplast. Not surprisingly, given its role as the critical chemical that enables plants to build food molecules, rubisco is the most abundant protein on Earth. Secondly is sugar creation. The newly built molecule immediately splits in 2, and each half is chemically modified. First, a phosphate group from ATP is added, and then each of the 2 molecules receives some high-energy electrons from NADPH. The result is 2 molecules of a 3-carbon sugar called glyceraldehyde 3-phosphate. For the net synthesis of 1 molecule of G3P, or glyceraldehyde 3-phosphate, from carbon in the atmosphere, 3 turns of the Calvin cycle are required to fix 3 molecules of carbon dioxide. However, some of the G3P molecules are combined to make the 6-carbon sugars glucose and fructose. These sugars can be used as fuel by the plant, enabling it to grow. They can also be used as fuel by animals, including humans, that eat the plant. Thirdly, we have regeneration. In regeneration, not all the G3P molecules are used to produce sugars. In the third and final phase of the Calvin cycle, some G3P molecules are rearranged to regenerate the original 5-carbon molecule. The rearrangement of G3P requires energy from ATP produced in the photo reactions of photosynthesis. Once the 5-carbon molecule is regenerated, the Calvin cycle can continue to fix carbon and produce molecules of G3P. The 3 turns of the Calvin cycle required to synthesize 1 molecule of G3P from carbon in the atmosphere consume 9 molecules of ATP and 6 molecules of NADPH generated in the photoreactions of photosynthesis. And that brings us to the take-home message of section 5.10. And that is, during the Calvin cycle, the synthesis part of photosynthesis, carbon from carbon dioxide in the atmosphere is attached to molecules in the stroma of chloroplasts. and sugars are built and molecules are regenerated to be used again in the Calvin cycle. The fixation, building, and regeneration processes consume energy from ATP and NADPH, the products of the photo part of photosynthesis. Moving on, Sudan, Ethiopia, India, Somalia. Many of the regions with the highest rates of starvation are also places with the hottest, driest climates. This is not a coincidence. These climate conditions present difficult challenges for sustaining agriculture, and in the absence of stable crop yields, food production is unpredictable and the risk of starvation is high. But evolutionary adaptations in some plants enable them to thrive in hot, dry conditions. When it gets too hot and dry, animals can seek coolness in the shade. Plants, however, are anchored in place. Consequently, plants in hot, dry climates lose significant amounts of water through evaporation. Evaporation is a problem for plants because water is essential to photosynthesis, growth, and the transport of nutrients. Without water, plants die. Moving on, to combat water loss through evaporations, plants can close their stomata. A stomata are the small pores usually located on the underside of leaves. These openings are the primary sites for gas exchange in plants. Carbon dioxide for photosynthesis enters, and oxygen generated as a byproduct in photosynthesis exits through them. When open, the stomata also allow water to evaporate from the plant. Closing their stomata solves one problem for plants, that is too much water evaporation, but creates another. With the stomata shut, oxygen from the photoreactions of photosynthesis cannot be released from the chloroplasts, and carbon dioxide cannot enter. And without carbon dioxide molecules for sugar production, the Calvin cycle cannot proceed. Plant growth comes to a standstill and crops will fail. Moving on, in some plants, including corn and sugarcane, a process has evolved that minimizes water loss but still enables the plants to make sugar when the weather is hot and dry. In the process called C4 photosynthesis, these plants add an extra set of steps to the usual process of photosynthesis known as C3 photosynthesis. In these steps, the plants use an enzyme that functions like an ultimate CO2 sticky tape. The enzyme has a tremendously strong attraction for carbon dioxide. It can find and bind carbon even when carbon dioxide concentration is very low. As a consequence, the plant's stomata can be opened just slightly, reducing evaporation and admitting a little CO2. In contrast, the enzyme rubisco functions poorly when CO2 is scarce, necessitating greater stomata opening. This adaptation has a catch, though. The extra steps in C4 photosynthesis require the plant to expend additional energy. Specifically, every time the plant takes up a molecule of carbon dioxide with the quote-unquote carbon dioxide sticky tape enzyme, the plant must use an extra molecule of ATP, which is required for the production of that enzyme. It is acceptable to pay this energy cost only when the climate is so hot and dry that the plant would otherwise have to close its stomata and completely shut down all sugar production. In a mild climate, however, plants using C4 photosynthesis would be outcompeted by the more efficient plants carrying out standard C3 photosynthesis. Not surprisingly, we see few C4 plants in the temperate regions of the world and little C4 photosynthesis among the photosynthetic organisms living in the oceans. Moving on, but in hot, dry regions, C4 plants, which include corn, sorghum, millets, sugarcane, and many grasses, are dominant and displace the C3 plants wherever both occur. With global climate change, most scientists predict a gradual expansion of the ranges over which C4 plants grow, while non-C4 plants, which make up the vast majority of plants, get pushed farther and farther away from the equator. And this is page 133 of your book, figure 5.26, and we can see the distribution of C3 and C4 plants globally. So that brings us back to the previous slide, and we see a third and similar method of carbon fixation called CAM. CAM stands for cressoluthian acid metabolism, and this type of photosynthesis is also found in hot, dry areas. In this method, used by many cacti, pineapples, and other fleshy, juicy plants, the plants close their stomata during hot, dry days. At night, they open the stomata and let CO2 into the leaves, where it binds temporarily to a holding molecule. During the day, the holding molecule gradually releases CO2, enabling photosynthesis to proceed while keeping the stomata closed to reduce water loss. However, I do need to point out a disadvantage of CAM photosynthesis. A disadvantage of CAM photosynthesis is that by completely closing their stomata during the day, CAM plants significantly reduce the total amount of carbon dioxide they can take in. As a consequence, they have much slower growth rates and cannot compete well with non-CAM plants under any conditions other than extreme dryness. C4 and CAM photosynthesis originally evolved because they made it possible for plants to grow better in the world's hot and dry regions. Today, researchers are now experimenting with these adaptations as a way to fight world hunger. They have introduced into rice plants several genes from corn that code for the C4 photosynthesis enzymes. Once in the rice, these genes increase the rice plant's ability to photosynthesize, leading to higher growth rates and food yields. So whether the addition of C4 photosynthesis enzymes will make it possible to grow new crops on a large scale in previously inhospitable environments is not certain. Early results suggest, however, that it is a promising approach. Okay, and that brings us to our take-home message for section 5.11. C4 and CAM photosynthesis are evolutionary adaptations at the biochemical level that, although more energetically expensive than C3 photosynthesis, allow plants in hot, dry climates to partly or fully close their stomatas and conserve water without shutting down photosynthesis. Moving on, food is fuel. And all the activities of life, growing, moving, reproducing, require fuel. Plants, most algae, and some bacteria obtain their fuel directly from the energy of sunlight, which they harness through photosynthesis. Less self-efficient, on the other hand, such as humans, alligators, and insects, those type of organisms must extract their energy that they need from the food they eat. This energy from photosynthetic organisms, either directly from eating plants or indirectly from eating animals that eat plants. Moving on, all living organisms, including plants, extract energy from the chemical bonds of molecules, which can be considered quote-unquote food, through a process called cellular respiration. So don't confuse cellular respiration with the act of breathing, which is also called respiration. Cellular respiration is a process that is a bit like photosynthesis in reverse. In photosynthesis, the energy of the sun is captured and used to build molecules of sugars such as glucose. In cellular respiration, plants and animals break down the chemical bonds of sugars and other energy-rich food molecules such as fats and proteins to release the energy that went into creating them. In humans, as in other animals, cellular respiration starts after we eat food, digest it, absorb the nutrient molecules into the bloodstream, and then deliver them to our cells that are located throughout our body. At this point, our cells begin to extract the energy stored in the bonds of the nutrient molecules. As energy is released, cells capture and store it in bonds of ATP molecules. Ultimately, when a nutrient molecule has been completely processed in the cell has used the nutrient molecule's stored energy along with oxygen to create a large number of high energy storing ATP molecules which supply energy to power the cell's activities, water, and carbon dioxide, which is exhaled into the atmosphere. This plentiful, readily available stored energy can then be tapped as needed. And that brings us to our take-home message for section 5.12. Living organisms extract energy through a process called cellular respiration, in which the bonds of energy-rich molecules are broken, releasing the energy that went into creating them. The process uses fuel and oxygen and yields ATP, water, and carbon dioxide. Moving on. To generate energy, fuels such as glucose and other carbohydrates, as well as proteins and lipids, after some preliminary modifications, are broken down into 3 steps: glycolysis, the citric acid cycle, and the electron transport chain. Moving on, glycolysis means the splitting or the lysis means to break, of sugar, sugar, so glycolysis, to break sugar. And this is the first step that all organisms take to capture energy from the breakdown of food molecules. For many single-celled organisms, this one step is sufficient to provide all the energy they need. Glycolysis comprises a sequence of 10 chemical reactions taking place in the cytosol of the cell, through which glucose is broken down to form 2 molecules of pyruvate. Glycolysis has 2 distinct phases: an uphill preparatory phase and a downhill payoff phase. So you have to spend money to make money, correct? You've heard that phrase before, right? That's an old urban legend or cliche or something like that. Similarly, before any energy can be extracted from glucose, some energy must be added to the molecule. This addition occurs during the uphill phase of glycolysis. The additional energy, which comes from ATP, destabilizes the glucose molecule, giving it a more reactive chemical structure and making it ripe for chemical breakdown. Once the glucose can be broken down chemically, the downhill phase payoff begins. During the payoff phase, as bonds in the sugar are broken and other lower energy bonds are formed, the energy released is quickly harnessed by the attachment of the phosphate groups to molecules of ADP to create energy-rich ATP molecules. In other energy-yielding steps of glycolysis, electrons originally from the glucose are transferred to NAD+ to become the high-energy electron carrier NADH. Later, in an electron transport chain in the mitochondria, this energy is converted into ATP. The net result of glycolysis is that each glucose molecule is broken down into 2 molecules of pyruvate. During this breakdown, some of the energy released from the breakdown is captured in the production of energy-rich ATP molecules and molecules of the high-energy electron carrier NADPH. 2 molecules of water are also produced during glycolysis. Glycolysis can proceed with or without oxygen. In other words, the reaction is either anaerobic or aerobic. In the absence of oxygen, and in many yeasts and bacteria, glycolysis is the only means for fueling activity. Because single-celled organisms have much lower energy needs, they can function solely on the yields of glycolysis. When oxygen is present, however, glycolysis isn't the end of the story. For many organisms, including humans, glycolysis is a springboard to further and much greater energy extraction. The additional energy payoffs come from the citric acid cycle and the electron transport chain. And that brings us to take-home message 5.13. Glycolysis is the initial phase in the process in which all living organisms harness energy from food molecules. Glycolysis occurs in a cell's cytosol and uses the energy released from breaking down chemical bonds in food molecules to produce the high-energy molecules ATP and NADPH. Moving on, cells could stop extracting energy when glycolysis ends, but they rarely do so, because that would be like leaving most of your meal on your plate. In glycolysis, only a small fraction of energy stored in sugar molecules is recovered and converted to ATP and NADPH. Cells get much more of an energy bang for their food buck in the steps following glycolysis, which occur in the mitochondria in the presence of oxygen. This is why mitochondria are considered ATP factories. In the mitochondria, the molecules produced from the breakdown of glucose during glycolysis are further broken down. The citric acid cycle, also known as the Krebs cycle, produces some additional molecules of ATP and, more importantly, they capture a huge amount of chemical energy by producing high-energy electron carriers. Moving on, before the citric acid cycle can begin, however, the end products of glycolysis, 2 molecules of pyruvate for every molecule of glucose used, must be modified. First, the pyruvate molecules move from the cytosol into the mitochondria, where they undergo 3 modifications and prepare them to be broken down into the citric acid cycle. In the first modification, each pyruvate molecule passes a pair of its high-energy electrons and a proton to the electron carrier molecule NAD+, building 2 molecules of NADH. In the second modification, a carbon atom and 2 oxygen atoms are removed from each pyruvate molecule and released as a carbon dioxide molecule. The carbon dioxide molecules diffuse out of the cell and eventually out of the organism. In humans, for example, these carbon dioxide molecules pass into the bloodstream and are transported to the lungs, from which they are eventually exhaled. In the third modification, which is the final step in the preparation for the citric acid cycle, a giant compound known as coenzyme A attaches itself to the remains of each pyruvate molecule, producing 2 molecules called acetyl-CoA. Each acetyl-CoA molecule is now ready to enter the citric acid cycle. Moving on, the citric acid cycle includes 8 separate steps, but our emphasis for our class will be on its 3 general outcomes. Firstly, a new molecule is formed. Acetyl-CoA adds its 2-carbon acetyl group to the starting material of the citric acid cycle. Then, a 4-carbon chemical called oxaloacetate, creating a 6-carbon molecule called citrate, is created. Secondly, high-energy electron carriers, NADH, are made and carbon dioxide is exhaled. The 6-carbon molecule then gives its electrons to NAD+ to make the high-energy electron carrier NADPH. And don't forget that the main purpose of the citric acid cycle is to capture energy. The 6-carbon molecule also releases 2 carbon atoms along with 4 oxygen atoms to form 2 carbon dioxide molecules. In mammals, including humans, this carbon dioxide is carried in the bloodstream to the lungs. And the third outcome is that the starting material of the citric acid cycle is reformed. And then ATP is generated and more high-energy electron carriers are formed. After the carbon dioxide is released, the remaining 4-carbon molecule is modified and rearranged to once again form oxaloacetate, the starting material of the citric acid cycle. In the process of this reorganization, 1 ATP molecule is generated and more electrons are passed to NAD+ and a molecule of FAD to form NADH and FADH subscript 2, respectively. Both are high-energy electron carriers. One oxaloacetate is reformed, and the cycle is ready to break down the second molecule of acetyl-CoA. 2 turns of the cycle are necessary to completely dismantle our original molecule of glucose. Moving on, now that we have seen the citric acid cycle in its entirety, let's trace the path of the 6 carbons in the original glucose molecule. We will see that in a sense, the carbon atoms plucked from the atmosphere to make sugar during photosynthesis have been exhaled back into the atmosphere as 6 molecules of carbon dioxide. First is glycolysis, the 6-carbon starting point. Glucose is broken down into 2 molecules of pyruvate. No carbons are removed. Secondly, we have the preparation for the citric acid cycle. In this step, 2 carbons are released. 2 pyruvate molecules are modified to enter the citric acid cycle and both lose a carbon atom, forming 2 molecules of carbon dioxide. Third is the citric acid cycle. The last 4 carbon atoms are released in the citric acid cycle. A total of 4 carbon atoms enter the citric acid cycle in the form of 2 molecules of acetyl-CoA, entering one at a time. For each turn of the citric acid cycle, 2 molecules of carbon dioxide are released. So the 2 final carbons are released into the atmosphere during the second turn of the wheel, and the 6 carbon atoms originally present in our molecule of glucose are no longer present in the organism. Now we've come full circle. In photosynthesis, carbon atoms from the atmosphere were used to build sugar molecules, which had energy stored in the bonds between their carbon, hydrogen, and oxygen atoms. In cellular respiration, the energy previously stored in the bonds of the sugar is converted into molecules of ATP, NADH, and FADH2. Carbon atoms from the sugar are exhaled back into the atmosphere as carbon dioxide, and water is produced. And that brings us to our take-home message for section 5.14. Much additional energy can be harvested by cells after glycolysis. First, pyruvate is chemically modified. Then, in the citric acid cycle, the modified pyruvate is broken down. This breakdown releases carbon into the atmosphere as CO2, and bonds are broken and capture some of the released energy in 2 ATP molecules and many high-energy electron carriers for every glucose molecule. Moving on, how do we finally get a big payoff of usable energy from our glucose molecule? In this energy held in the high-energy electron carriers, NADH and FADH2, formed in glycolysis and the citric acid cycle, that ultimately generates the largest amount of usable energy as ATP. In fact, almost 90% of the energy payoff from the molecule of glucose is harvested in the final step of cellular respiration when the electrons from NADH and FADH2 move along an electron transport chain. This process, like the citric acid cycle, takes place in the mitochondria. In a manner similar to that seen in the chloroplast during photosynthesis, mitochondria convert kinetic energy from electrons into potential energy, a concentration gradient of protons. 2 structural features of mitochondria are essential to their ability to harness energy from molecules. First, mitochondria have a bag within a bag structure. Consequently, material inside the mitochondria can lie in 1, the intermembrane space, which is outside the inner bag, or 2, the mitochondrial matrix, which is inside the inner bag. With 2 distinct regions separated by a membrane, the mitochondrion can create higher concentrations of molecules in one area or the other, producing a concentration gradient. Secondly, the inner bag of the mitochondrion is studded with molecules, mostly electron carriers, that are sequentially arranged as a chain. This arrangement allows the molecules to hand off their electrons in an orderly sequence, and this enables them to harness the electron's energy efficiently. Moving on, In step 1 of the electron transport chain, it will begin with NADH and FADH2 in the mitochondrial matrix. In other words, inside the inner bag, moving to the membrane. There, the high-energy electrons they carry are transferred to molecules embedded within the membrane. After they donate their electrons, the molecules that remain, NAD plus and FAD, are recycled back into the citric acid cycle. The membrane-embedded molecules pass the electrons to the next carrier, which passes the electrons to the next, and so on. At each handoff, energy is released. Thus, as electrons move from one carrier to another, they lose energy. At the end of the chain, the lower energy electrons are handed off to oxygen, which then combines with free hydrogen ions in the mitochondrial fluid to form water. Really, that's why you breathe, because oxygen is the final electron acceptor. So as shown in step 2 of figure 5.34, which is on page 140 of your book, most of the energy released at each handoff from one electron carrier to another in the electron transport chain is used to pump protons or hydrogen ions from the mitochondrial matrix across the membrane and into the intermembrane space. As more protons are pumped across the membrane and packed into the intermembrane space, a concentration gradient is created. This gradient represents a significant source of potential energy. Moving on, this description may seem familiar. For instance, in chloroplasts, during photosynthesis, many protons are pumped from the stroma outside the thylakoid sacs to the inside of the thylakoids. We liken this potential energy to the potential energy of water in an elevated tower, which can be released with great force. Similarly, in step 4 of the mitochondrial electron transport chain, the protons pumped into the intermembrane space rush back into the mitochondrial matrix through channels in the inner mitochondrial membrane. And as the protons pass through, the force of their flow fuels the attachment of free-floating phosphate groups to ADP to produce ATP. In the end, the complete dismantling of one molecule of glucose yields about 32 molecules of ATP. Here's an approximate accounting. Glycolysis generates 2 ATP and 2 NADH. The oxidation of pyruvate to acetyl-CoA generates 2 NADH, and the citric acid cycle generates 2 ATP, 6 NADH, and 2 FADH. In the electron transport chain, those 10 NADH generate about 25 ATP, and the 2 FADH2 generate about 3 ATP, bringing the overall ATP total to 32. An exact accounting isn't possible for a variety of reasons. A cell does not always utilize NADH and FADH2 solely for the production of ATP. Moving on, although we have examined just the breakdown of glucose, humans and all other animals are able to harvest energy from a variety of food sources. Whether a meal contains carbohydrates, lipids, proteins, or some combination thereof, the nutrients are chemically modified in some preliminary steps and then fed into one of the intermediate steps in glycolysis or the citric acid cycle to furnish usable energy for the organism. And that brings us to our take-home message for section 5.15. The largest energy payoff of cellular respiration comes as electrons from the NADH and the FADH2 produced during glycolysis and the citric acid cycle move along the electron transport chain. As the electrons are passed from one carrier to another, energy is released, pumping protons into the mitochondrial intermembrane space. As the protons rush back into the mitochondrial matrix, the force of their flow fuels the production of large amounts of ATP. Moving on, every beer brewery, the entire wine industry, and all distilleries of vodka, tequila, and other alcoholic beverages owe their existence to microscopic yeast cells scrambling to break down their food for energy under stressful conditions. To better understand how yeast metabolism produces alcohol, it helps to begin by investigating what happens when humans and other animals try to metabolize energy from sugar molecules under some stressful conditions. Moving on, with rapid, strenuous exertion, our bodies soon fall behind in delivering oxygen from the lungs to the bloodstream, to the cells, and finally to the mitochondria. Remember what I said earlier, oxygen deficiency limits the rate at which the mitochondria can break down fuel and produce ATP. This slowdown occurs because the electron transport chain requires oxygen as the final electron acceptor of all electrons generated during glycolysis and the citric acid cycle. If oxygen is in short supply, the electrons from NADH and FADH2 have nowhere to go. This whole process of cellular respiration can grind to a stop. Organisms don't let this interruption last long, though. Most have a backup method for breaking down sugar. Moving on, in animals, there is an electron acceptor for the NADH electrons in the absence of oxygen, and that is pyruvate. Pyruvate is the end product of glycolysis. When pyruvate accepts the electrons, it forms lactic acid. Once the NADH gives up its electrons, NAD plus is generated and glycolysis can continue. But as lactic acid builds up, it causes a burning feeling in our muscles. The next day, muscle soreness is not due to this acid buildup, which goes away in a matter of minutes or hours, but rather to the damage to the muscle fibers. They break down a bit before growing stronger. It's not ideal, but to escape a predator or exercise strenuously, the two ATP molecules generated from each glucose molecule during glycolysis, which can serve as an immediate energy source, are better than nothing. Like humans, yeasts typically use oxygen during their breakdown of food, and like humans, they have a backup method when oxygen is not available. But yeast cells use a different electron acceptor, and the resulting reaction leads to the production of all drinking alcohol. After glycolysis, in these single-celled organisms, pyruvate is usually converted to a molecule called acetaldehyde, and this releases bubbles of CO2 in the process. In the absence of oxygen, acetaldehyde accepts the electrons released from NADH, allowing glycolysis to resume. Acetaldehyde's acceptance of NADH's electrons results in the production of ethanol, the molecule that gives beer, wine, and spirits their kick. So, class, there's actually an easier way to think about this. When you drink alcohol, you're basically drinking yeast shit. Moving on, fermentation is the process by which cells obtain energy in the absence of oxygen. It occurs when, following glycolysis, alternative molecules are used as electron acceptors. Interestingly, although ethanol is always the alcohol produced by fermentation, the flavor of the output of fermentation depends on the source of the sugar metabolized by the yeast. Fruits, vegetables, and grains all give different results. If the sugar comes from grapes, wine is produced. If the sugar comes from germinating barley plant, beer is produced. Potatoes are the sugar source typically used to produce vodka. Many non-alcoholic products, including yogurt and soy sauce, are produced by fermentation. Because yeasts prefer the more efficient process of aerobic respiration, they produce alcohol only in the absence of oxygen, in other words, anaerobic. Fermentation tanks used in producing wine, beer, and other spirits are built to keep oxygen out so that yeast cells are forced to use their backup pathway of fermentation. And that brings us to our take-home message for section 5.17, and that is when oxygen is available, yeast cells resort to fermentation. In fermentation, they use a different electron acceptor, acetaldehyde, and in the process, they generate ethanol. Ethanol is the alcohol in beer, wine, and other spirits.